Hey, welcome to a new experiment. In lieu of a poster session, I thought I'd give you a short presentation about how we've used a new resource we've developed in the lab in order to try to accelerate biological discovery. So this video is going to demonstrate a scientific discovery that we think we've made using this platform. In a second video, I will perform a short walkthrough of this resource that we've generated for you then to waste some time of your day today on. So let's dive right into it. We've developed a resource called Data-Driven Hypothesis. This is a web app that we built in order to try to accelerate biological discovery. So let me tell you a little bit about it. Fortunately, new tools in data science are making it easier than ever before to make discovery from data. And when we say data science, I want to try to explain what we mean. Data science is a term that's thrown around a lot. When I say data science, what I mean is a series of tools that allow us to extract information from data in order to try to learn something from it. So these tools are what we call data science. There's a lot of different data science tools. One of the things that's required before diving into data science is to have some high quality data. Some of the data that I've used to generate this resource is from the Broad Institute. The Broad has a fantastic platform where they are systematically profiling gene expression and gene dependency data in order to try to learn something about cancer precision medicine. We can use these data to extract patterns to try to learn something about the genes that we're interested in. So what does this look like? The Broad starts by performing whole genome functional screening, essentially knocking out every single gene in the genome from hundreds, and in fact, almost up to 1,000 different cell lines, a very broad thing to do. We can then take these data and we can ask which cell lines care about which genes. We can assign these what's called a dependency score. This dependency score will then score how much the cell cares about not having that gene. A score below -1 means a dependency score where the gene is considered essential. If we knock out this gene of a cell line, it is absolutely required for growth and the cells stop growing. On the flip side, we also have genes that when knocked out, the cells are going to gain fitness or grow better. Sometimes we call these tumor suppressors. So this is the premise and the raw data here of the dependency scores for all genes across the human genome for approximately 1,000 cell lines. Here's a simple example illustrating how we can find relationships between genes that behave similarly. So what you're looking at is the nucleotide biosynthetic pathway. One key enzyme is an enzyme called CAD or CAD. If we knock out CAD, what happens is the cells are going to care deeply about this. They are going to absolutely require CAD because without this, they cannot make nucleotides, and nucleotides are important for DNA repair and replication. So we can look and see which cells care deeply about CAD. And it turns out we have several. If we then ask the question, which cells share a similar pattern of behavior to the CAD knockout, we can find a handful of other genes that behave similarly. This process then allows us to infer something about nucleotide biosynthetic genes. So back to the schematic. If our cells don't have CAD, they're not going to have nucleotides, and a subset of these cells will die. Now, it doesn't matter how you knock out nucleotide biosynthesis. If the cells don't have nucleotides because you knock out another gene in the pathway, then the cells will also die. But again, and here's the key idea, only a subset of the cells care about nucleotide biosynthesis. And because of the heterogeneity in this pattern, we can then look and see which genes are behaving like which other genes. And when it turns out we look at genes with similar patterns of dependency as CAD, it's all of the genes in the nucleotide biosynthetic pathways. In fact, all of the ones in orange are the ones that we found. Now, one of the key activities in biomedical research is identifying new functions for genes and proteins. So how do we do that? What's the very first experiment you would do? We could try to map human gene functions to patterns of similarity to other genes. So for the rest of my time here, I want to give you a short test case. It's a project that we're actively working on, It's very new. We don't have all of the answers, but I'm excited to share it with you today. So we have looked across several metabolic genes, trying to identify new functions for these. One of the genes we've looked at is a gene called succinate dehydrogenase. This, of course, is a multi-subunit complex involved in oxidative phosphorylation, as well as the TCA cycle. We've looked specifically at succinate dehydrogenase, subunit B, and this has some interesting patterns that emerged. When we look and see which cells care about succinate dehydrogenase B, we find several. Importantly, there's enough variability in the pattern here that we can identify genes that behave similarly. When we look at the distribution, we can see that there are several cells that don't care about succinate dehydrogenase B, but importantly, a set of cells do, and this encourages us to look a little bit further. Now, when we look at genes that are behaving similarly when knocked out to succinate dehydrogenase B, we see the expected players, SDHA, C, D. If a cell line is going to care about succinate dehydrogenase activity, and we have 4 different subunits that are absolutely required, then it makes sense if we knock out succinate dehydrogenase B, some cells care, and those are the same cells that care if you don't have A, C, or D. Now, the thing that was surprising was that the next sets of genes that emerged from this were, in fact, nucleotide biosynthetic genes, the genes I just told you about. So this is surprising because succinate dehydrogenase B has no business being associated with or having similar dependency patterns as genes involved in nucleotide biosynthesis. So what this could mean is that if a cell lacks a nucleotide biosynthetic gene, it is not going to be able to make nucleotides. But these data also imply that if a cell lacks succinate dehydrogenase B, it will also not be able to make nucleotides. And this now sounds like something that would be ripe for discovery. So the first thing that we wanted to do was to test out this hypothesis. We can see from a different view that this network of connections of succinate dehydrogenase B, as well as the nucleotide biosynthetic genes, are highly interrelated. So it leads to the hypothesis then, is succinate dehydrogenase playing a role in nucleotide biosynthesis? The first thing that we can do is just measure steady state nucleotides. One of the challenges with this project is that there are several nucleotides. There are also even more nucleotide intermediates. So we started with an unbiased approach, just looking at steady state levels of nucleotides from cells where we knocked down SDHB, SDHA, or 2 other cell lines that didn't contain known enzymes involved in the nucleotide biosynthetic pathway. And this is CAD and ADSL. So what does this look like? You can see up here 2 things I want to point out. The first is that if we take out succinate dehydrogenase, levels of succinate are way higher than if we had succinate dehydrogenase. And again, this serves as a nice positive control because we now don't contain the complex that is required to consume succinate. If we look at the metabolites that are lower in the absence of SDH, we can see CTP, sucralfate acid, and IMP. So IMP and CTP are known intermediates in nucleotide biosynthesis. And so this was encouraging and suggested that there could be something there. So we looked a little bit further. Diving deeper into a statistical analysis revealed a few other metabolites that were different between our conditions. We can see that UDP and adenylosuccinate are also lower in the absence of SDHB compared to our scramble control. We can also see that UDP and adenylosuccinate are lower in the absence of SDHB compared to our scramble control. Because cells are going to be finely tuned to the levels of nucleotides, we wanted to dive into this further and ask if nucleotide biosynthesis, not just nucleotide levels, could be different in the absence of SDHB. To do this, we traced stable isotope labeled carbon and glucose into nucleotide pools. So the carbon from glucose is going to end up in the ribose ring. This is a way that we can trace the carbon from glucose into ribose and ultimately into steady state levels of nucleotides. So in collaboration with Ann Lee at Johns Hopkins University, that's exactly what we did. Now, if you recall, CTP was one of the metabolites that we measured, and our bulk metabolite analysis that was lower in the absence of SDHB, and we would look at our steady state carbon flux into this metabolite, we could see it's also lower. We also see that flux into adenosine is lower in the absence of SDHB. So altogether, when we take all of these metabolomics data and we put them into a metabolite set enrichment analysis, we can see that the metabolites that are most significantly different in the pathways that those metabolites represent are pyrimidine metabolism. And so while we don't yet know the mechanism of how SDHB is influencing these levels, it looks like the data from our resource predicted a role for SDHB that then our experimental evidence at the bench was able to validate. So how does SDHB influence nucleotide biosynthesis? SDHB sits at this important nexus. It's got roles in oxidative phosphorylation. It's got roles in the TCA cycle. It also has other roles that we're trying to disentangle to understand how it's influencing nucleotide biosynthesis. So we don't yet have the answer, but we're working hard to try to understand this. Now, I'd just like to finish up by acknowledging the funding sources, by acknowledging the members in the lab who are working hard on this project, and I'd like to thank you for your attention for making it this far. If you're interested in this resource that we've developed and want to try to develop some data-driven hypotheses for your project, then go ahead and check out this next video where I'll give you a short walkthrough and how to use it. Thanks for watching.